Hi, everyone. Welcome back to the ALBA Child Health and Nutrition podcast. Here we interview scientists, doctors, and experts in gut microbiome research, and especially on child health and nutrition. And today we have an amazing guest, Dr. Henrik Roager, if I pronounce it.
That's absolutely fine.
From the University of Copenhagen, who is researcher in microbiome science and a leading expert also in nutrition. Henrik, I'm really excited to have you today. Maybe great to get an introduction on you first.
Yeah, thank you. Yeah, I'm an associate professor and I'm leading a research group that is dedicated to understand sort of the interactions between what we eat, the microbes in our gut and our health. And I've been involved in this research for more than 10, 12 years. So I've sort of seen the field evolve during the last decade, and I find it incredibly exciting to work in this research field.
And you've also put together the Copenhagen Gut Microbiome Hub, which is a group of researchers that come together every year, and in general, a community in gut microbiome research. What makes you excited about gut microbiome research?
Yeah, it's true. We established this hub. I did that together with other leading researchers because we wanted to bring the community closer together. Because I think one of the things that makes me really excited by this field is that you need to work across disciplines. So we need people that are experts in microbiology, nutrition, immunology, bioinformatics to ultimately understand what happens in our gut and how it relates to our health. And I think by bringing the community closer, my hope is to basically facilitate more interactions, more collaboration with the ultimate goal of advancing our science and also making it more fun.
That's fantastic. I was there at the first symposium and there were so many interesting studies and publications and presentations from many different areas. How did you get started on this topic? How did you get into gut microbiome science?
I guess it was a bit of a coincidence. When I was a student, I saw that there was a talk by a professor on probiotics and prebiotics, and I was very interested in microbiology. And when I realized that microbes can actually be beneficial, and it's not only bad bacteria we should be concerned about, but also the beneficial microbes, that sort of gave me interest in the field. And I got to work on a bachelor project on infant microbes and a protein in breast milk and whether this protein could affect the bacteria. And then later I did my master thesis as well, also on the gut microbiome, but then in relation to adults and diet. And that just fueled my interest. And then I was lucky to be at the right place to also do a PhD. And one thing took the other, and I couldn't stop doing research because simply I enjoyed it too much. Yeah.
And now it's a very exciting time for gut microbiome research because we went from maybe hundreds, few thousands of publications every year to I think it was 20,000 last year. So it's a time where knowledge is increasing a lot. What do you think that gut microbiome research will help us do in the future?
I agree. It's knowledge is increasing a lot, but I also think at the same time noise is also increasing. So it's getting more and more difficult to also navigate the field and figuring out what is actually advancement and what is not advancement. But what I really hope that we will be able to do in the future is, first of all, to understand our digestion better. So basically understand why do we respond differently to the same diet. Also to understand sort of how is their physiology and their digestive process affected by our microbes in our gut, and what does it mean for later health? I mean, there's a huge potential to understand digestion and the GI tract much better. And here I think understanding how we can affect our health through the gut is super interesting because there is so much physiology going on, or so much metabolic processes, the nervous system, the immune system, the endoendocrine system, which is the hormonal system, which is somehow regulated through the intestine. I think that holds a great potential that I hope we can be much better in understanding.
It's very complex.
Yeah, very much.
And when we talk about the microbiome, we talk about a lot of things. Actually, let's start from the basics. What is the gut microbiome?
So when we say the gut microbiome, we typically refer to all the microorganisms living in the gut. That includes bacteria, fungi, viruses, all microorganisms we can think of, and we term that their gut microbiome. Strictly speaking, you can also talk about the microbiome as all the genes that the microbes encode in the gut, but generally I refer to all the microorganisms in the gut when I talk about the gut microbiome.
And it seems that the gut microbiome is linked to health or important for our health. What do we know about that in general terms?
So in general terms, we know that the microbiome is important for digestion of undigestible fibres, for example. So we know that their fermentation in the large intestine is largely driven by the microbes, which have some enzymes that make them able to degrade all the food components that our own digestive enzymes can't digest, basically. So we know that that results in additional energy for our body and also for to utilizing the food we eat. Then we also know that microbes produce certain vitamins, and that way can affect our health. And we know that the microbes is also able to regulate and affect the immune system in the intestine, for example. Then it's also interesting that the composition of microbes somehow relates to our environment, to our diets, to the way we live. So it's also somehow reflecting our lifestyle. So you can also basically, by reading the microbiome a bit, you can somehow get an idea of lifestyle or diet, basically. And I think that's interesting as well. So it's basically responding to what we expose it to.
And when we say the microbiome affects digestion and how we extract energy from food, does it also mean it could affect weight?
Yeah, so we know from animal experiments that the microbes can affect weight. That's for sure. That has been nicely demonstrated that if you take microbes from an obese donor and transplant into what we call germ-free mice, so mice that are sterile, then these mice that get the microbes from the obese donor, they will gain more weight compared to mice that get microbes from a lean donor. So that has been shown that the microbes this way is a causal factor that could contribute to obesity. In adults or in humans, it's less clear. So we have hypotheses on the same lines that the microbes could as well affect energy excretion or energy extraction in the intestine. We also have the ideas that microbes could affect secretion of appetite hormones in the intestine that maybe could regulate appetite as well. But the evidence from humans is less clear.
Why is that? Why are the experiments different, or what makes these experiments different?
So I think the key difference is that when you do experiments in animals, you can really control the diet, you can control basically the experiment. When you do experiments with humans, we are per definition different. So we eat differently, we have different health trajectories. So it's simply more challenging. Also because typically when you do an intervention in humans, you would also realize that we respond very different. And that just makes it more complicated to consistently show an effect of a given intervention.
Yeah. And there are more and more articles in the press also linking the gut microbiome to the brain. What do we know about that?
Yeah, so I think most people have sort of the immediate experience that if you are stressed or you need to go to an exam or to a job interview, then often that results in a feeling of or urge to go to the toilet. So we have a clear feeling that the brain communicates to the gut. But the other communication from the gut to the brain is also likely to happen because we have nerves located all around the intestine that signals to the brain. So the brain is somehow monitoring what goes on in their gut with the key purpose of ensuring that digestion is as efficient as possible, and to ensure that the food passes through the intestine for absorption, but later for excretion also. But there are studies suggesting that if you have problems with your digestion, then your risk of getting depression is higher. So that we see associations between having problems with the GI system and then depression. So there is a clear indication that the gut and the brain is communicating, and that's why we think that regular bowel habits is really key also for mental well-being simply. So there's a lot of interest in that, but that's also super, super complicated.
Yeah, that's really, really interesting.
Yeah.
And why is that? What do these microbes do in our gut that can affect the brain?
So there are different ways we speculate that microbes affect the brain. One is via their nervous system, so via production of small molecules that can interact with the nervous system. Another way is that their microbes may affect the production of certain appetite hormones that are formed in the intestine that we know can affect nervous system as well, or maybe even circulate via the blood to the brain. And then we also have the idea that microbes can affect the immune system in the intestine. And we know that immune cells from the intestine do travel to the rest of the body, including the brain. So if your immune system is affected in the intestine, these immune cells could eventually end up in the brain. And depending on their programming or state, that could have different impacts in the brain as well. But I must say, this evidence is mainly from animal models, and it's at this point in time still a lot of hypothesis, and we are not at this stage where we can actually, in humans, manipulate the brain via the gut.
And what about the immune system? What do we know about the gut microbiome and how it affects the immune system?
So I think one has to realize that the intestine and the microbes are normally kept separated from our own epithelial cells, so the cells in the intestine that sort of protects us from the bacteria. So normally there is a barrier function there that keeps things separated in the intestine. But if that's broken down or impaired, then microbes can get in contact with our intestinal cells and induce inflammation simply. And the immune system is there, first of all, to fight potential infections and making sure that bacteria don't get into their body or into the blood. So that's very important. But we also think that a large proportion of the immune system is actually located below the intestinal cells and waiting there just to make sure nothing passes in, but maybe also being affected by different signaling molecules that microbes can produce that can pass over the intestinal barrier and enter into the area where the immune cells are present. So although the bacteria may not pass over, then the bacteria might still, via the production of small molecules, affect the immune system in the intestine. And that could potentially affect which kind of immune cells do we have and how would they respond to a given challenge.
And the microbiome can affect the immune system in a positive or negative way, right?
Yeah,
so
both ways.
Yeah, both ways.
Yeah.
So you said it could actually produce inflammation or cause inflammation, but also regulate the immune system in a good way.
Yeah, exactly. And it's always a matter of balance and keeping things in place. So that's really key for health. It's to keep things in a steady state or a stable situation. And
Exactly. And that's one of the things that I find sometimes so hard to explain about the gut microbiome, because we're so used to categorizing things into good and bad.
Yeah.
And bacteria and microbes can be good and can be bad, some more than others. But a lot of the time it's more about the ecosystem and an equilibrium also with the rest of the organs and our body.
Yeah, and it's often a matter of their circumstances as well. You can have a family, if you think about a well-functioning family, that works nicely in a certain situation, but then if you put certain stressors in there, then things can just blow up. And in the same way in the intestine, where you have a family of microbes that are all happy and thriving, well, that might be true until a certain period where, let's say, antibiotics comes in, kills off half of the bacterial family, and it creates a new uncertainty. And in this situation, you have a risk that a certain family member might take up a too dominating position and suddenly start enforcing bad things on the rest of the family. So it always depends on their circumstances. And
it also seems in the same way that many systems in our body or organs affect each other. So the gut affects the immune system, the brain, also the skin, other kind of systems in our body. What do we know about other things besides brain, metabolism, and immune system?
I don't know how to answer that.
It's a difficult question.
Yeah. Well, I think we have to realize that our body is incredible, complicated, and beautiful at the same way. So things are not, we cannot study one organ in isolation, right? Everything is somehow connected. And so if one organ is suffering, well, the whole body is suffering, right? So that's also why problems with the digestive system could have implications elsewhere, whether it manifests on the skin or in your stress level or whatever, might depend from individual to individual.
Yeah, that's a very good way to put it, actually.
Yeah.
Yeah, no, it's very, very interesting. And what do we know about the association between the gut microbiome and health conditions? There are some that have been associated in a stronger way than others.
So I think if you look at their studies that have been done in humans, most of the findings are sort of association studies where we can see that a poor lifestyle is linked to a more poor microbiome. We can see that if you don't eat a lot of dietary fibres, then that sometimes is associated with a less diverse microbiome. But whether their microbiome is then their direct link to a poor health as well is more questionable. We don't know always whether it's their lifestyle per se that's the driving force for a poor health state, or whether it's mediated via the microbes in the gut. And probably it's a combination. But when we do the human trials, it's often very difficult to disentangle what is causing what. Because the microbes will respond to changes in diet, but eating a healthy diet would also in itself have a direct impact on our health because we get all the nutrients, the vitamins, all we need for our body through the diet. So that in itself is a very important factor, of course. But part of it could also be happening through their intestine or through their microbes in the intestines. Yeah.
Right. A lot of people, sometimes when they hear that there's only association studies between the microbiome and health, they say, well, we don't know anything yet, so it doesn't really matter what I eat. But as you say, the diet itself has also been linked to health, and we have guidelines also.
We have very good reasons for eating a diverse diet full of vegetables, fruits, and fiber, making sure that you get different types of foods. There's really good evidence for that. I think where we need to be a little more clever is to understand why is it that we respond differently to the same kind of diet. And that's where I think the microbes and digestive system comes in, because that could provide a sort of an explanation for why some people feel bloated on a certain vegetables where others don't feel bloated on the vegetable.
Yep.
And that's where we can maybe help people feel better on the given diet.
And the gut microbiome seems to be even more important in the first years of life in children. Why is that?
So when children are born, they are basically sterile in their intestine. So that means that they will encounter the world through the intestine. And one thing I think is mind-blowing to think about is that their surface area of the intestine it's much bigger than the surface area of the skin. So that means that the children that face the world, they're actually primarily facing their microbes in the world through the intestine, because their surface area of the intestine is much bigger.
Wow.
So that means that so the children are really have to encounter and learn to recognize or different microorganisms that they will encounter via the intestine. And since the intestine is sterile once they're born, it also means that the intestine will gradually be colonized by the microbes that the children are being exposed to, that comes from the environment and from their breast milk, for example. And then slowly their children's intestine is being seeded and their microbes start to colonize. And since it's an ecosystem that needs to establish, it means it's also a period where there's a lot of changes and dynamics happening. So it's not a stable ecosystem. It's gradually changing and developing over time. And this development depends largely on their environmental exposure, but also by the nutrition, the feeding, basically, during the first months of life. And one thing more to think about is that in life, we don't have a larger change in our diet than from when we go from milk to solid foods. And that's a really drastic dietary change if you think about it. And since microbes respond to what we eat, then it's also a really a period where, the microbes will change dramatically from the period from milk to solid food. So this is a really a period where the microbes develop and changes a lot. And that happens in parallel with a time where the immune system is also developing and the child is growing dramatically, if you think about it. So it's really an important period for development in terms of microbes, immune system, nervous system, and growth in general. So that's why we think it's such an important window.
That's such an interesting way to see it. You know, how you explain that a child is encountering the world through their gut. That's very true. And we never think about it that way. And if we take it from the start, how important is birth and the way a child is born for their gut microbiome?
Yeah, so we know that children that are born vaginal compared to C-section have a different microbiome in the first weeks and months of life. And that's simply because that children born by C-section, they are exposed to different microbes than the children born vaginally.
Yeah.
So vaginally born children would typically have more bacteria that are seeded directly from the mother's vagina and her feces, basically. So that's why in the first weeks and months of life, we do see this difference of according to birth mode, basically. And that has also been associated with a higher risk of certain immune-related diseases later in life. So we think that part of this higher risk could be related to a different seeding in early life.
Which kind of conditions are associated with C-section?
Well, I think it's asthma and allergy in particular that seems to be where the risk is higher with C-section.
Yeah.
The underlying factor driving this increased risk is not fully understood. But the hypothesis is that it could be due to the early seeding of the infant's intestine. That could be part of the explanation. So that's also why that researchers are now doing experiments with trying to seed children that are born by C-section and seeding them with the mother's vaginal microbes or fecal microbes right after birth to see whether that would have implications in a beneficial way later in life when it comes to allergies, for example.
And then you mentioned that feeding is also very important.
Yeah.
Breastfeeding versus formula feeding. What do we know about that?
So we know that breastfeeding is a really, really important factor for shaping the gut microbiome in early life. And one of the main reasons being that in breast milk, the third largest component is a special group of carbohydrates, which we call human milk oligosaccharides. And what's special about these carbohydrates is that their child's own digestive enzymes cannot degrade them. That means that they pass on until the colon, where we have, or the child has, bacteria that can digest these human milk oligosaccharides. And in this way, you can say that breast milk selectively promotes certain bacteria in the intestine of the child. So it seems to be a built-in feature of the breast milk that you basically select for certain beneficial microbes in the child's intestine. And I think that's kind of mind-blowing. And it also means that these bacteria, which are called Bifidobacteria, they can dominate a breastfed child up to 90% of the infant gut microbiome, which is very unusual. You will never see that in adults. So that's really a unique thing for infants.
And these bacteria seem to be beneficial for children and associated to multiple health benefits. What do we know about it?
Yeah, so we generally have this idea that bifidobacteria are important and beneficial. And one of the things being that they can produce certain acids, so lactate and acetate, which lowers their pH in the intestine. And by lowering the pH in the intestine, you basically hinders other pathogenic bacteria to establish and colonize. So one of the mechanisms is really that it creates an environment in the intestine that keeps other bacteria away. So that's one way. Another way is that we think that they're also producing certain molecules that could stimulate the immune system in a beneficial way.
Yeah, so it seems like the breast milk contains some compounds that feed these good bacteria.
Yeah.
And about the bifidobacteria, there's also different types, right? And you had a very interesting discovery about what they produce.
Yeah, so in one of our research studies, we found that certain bifidobacteria are able to produce some molecules that we call aromatic lactic acids. But it's not all bifidobacteria. But the bifidobacteria that seems to be able to produce them are exactly those that are good at utilizing these human milk oligosaccharides. And what we think the aromatic lactic acids are doing is actually affecting immune cells, so shaping the immune system in early life. So what we can put together so far is that human milk oligosaccharides that you find in breast milk promotes certain types of bifidobacteria, and these certain types of bifidobacteria produce molecules that can affect immune function. So this could be one way by which these bacteria could be beneficial in early life. We still need to sort of do the final experiment proving that it actually happens this way in an infant cohort, you can say, but at least this is what we think at this point in time.
And some studies have associated higher bifidobacteria to a lower risk of allergy, for example, or other immune-related conditions.
Yeah. And I think what's difficult maybe to understand sometimes is that, yes, you might see that bifidobacteria is associated to lower allergy, but it's somehow very often difficult to understand. Is it the breastfeeding, the breast milk per se, or is it the bifidobacteria that makes a difference? Because these two often goes together. So it can be a little hard to finally conclude on what is their causal factor here.
What are the next steps then in this kind of research? And you have an amazing cohort study, the motility study in Denmark. What are your next avenues in this field?
Yeah, so one of the things we have realized is that if you study the gut of infants, you realize that we are all widely different also in early life. And that's why we have decided to do an infant cohort study where we follow children's development over time. So we have had parents collecting poo samples every second week for a year from birth to 1 year of age. And the idea is basically to be able to track over time how their microbiome develops depending on their diet, but also to understand how their development of the microbiome relates to bowel habits in early life. So that is one of our key interests, that is to understand bowel habits, but it's also to understand how microbes and their molecules relate to immune development in early life. And this is something that we hope to understand better in the future.
It also seems that some studies have linked, for example, colic or bowel function or gut or motility to, for example, immune system conditions and other conditions. Could there be a link between these different classes of conditions?
Yes, possibly there could be a link. Right now, it's a black box. We know very little about what's going on in early life. We know that bowel habits are highly variable among children. So some infants have diapers 7 times a day. Others, they have one every second week. And if you ask a medical doctor, he will say that that's perfectly normal, no problem. And yes, it might not be a problem, but we are still curious to understand why is there this huge variability in terms of defecation patterns, basically. And we know that so we have infantile colic, which is kind of a broad term used to define when children cry a lot over a longer period of time. The underlying reasons, that can probably be many, but we think one of the underlying reasons could be a microbiome that is somehow either producing a lot of gases or producing certain molecules affecting the immune system in a certain way, or that the children are simply constipated. But that, I mean, there's so much to be discovered here. But we hope to understand better how these bowel habits relate to the microbes, because it would be fantastic if we could give better advice to parents, relieve stress for the child and the parents. So that's the hope for the future.
And things are very variable between people, but also over time.
Yes,
right?
Definitely.
They change rapidly, and what happened 1 month ago is different 1 month later.
Yeah.
So it seems like there's a lot happening in that time. So it's great that you have those frequent samples as well.
Yeah.
Because then we'll have like a tighter view of what happens.
Yeah. Yeah, and one of the great things I hope we'll see is that, especially when the children go from milk to solid foods, that transition can, in some children, go really fast, and in others take a really long time. So it really depends on the child and the parents how fast the children transition from milk to solid foods. And most studies conducted today have asked for whether child breastfed, formula fed, or introduced to solid food. Yes, no. And the problem is that one thing is you introduce solid food, but it doesn't mean that your child is necessarily on a solid food for a larger extent. So with our study, we'll have a much better understanding of to what extent the child has transitioned to the solid food, because we are really in great detail following the transition from every second week over time with a questionnaire on the diet basically.
And what happens with the introduction of solids? So we talked about what happens before. There's breast milk or formula feeding, and then we have the introduction of solids. How does the gut microbiome change then?
Yeah, so I think the key thing is really that with introduction of solid foods, you introduce more substrates for the microbes. So that means ultimately you would have more different microbes that can utilize the different dietary substrates that you introduce to the system. So ultimately, you would over time see a gradual increase in bacterial diversity, because you basically feed more different bacteria, whereas as long as the child is mainly breastfed, it would be dominated by the Bifidobacteria that has a competitive advantage over the other microbes because it's so superior in utilizing these human milk oligosaccharides. And what we actually also see is that it's actually more when their children are not breastfed anymore that we really see a rapid increase in dietary diversity, in bacterial diversity. Until as long as the child is mainly breastfed, the diversity is still going to be rather low because breastfeeding has such a big influence on the microbiome.
And microbial diversity, so how many bacteria are present in the gut, that seems to be an indicator of health, or it has been associated to health. What do we know there?
So here we need to be a little Exact. Because actually low diversity during breastfeeding is probably a good thing. Whereas later when the child is reaching 9, 12 months, there at that point in time, we want to see a shift from low diversity community to a high diversity community. And the hypothesis is that a more diverse community would give a more stable ecosystem and allow for a microbiome that is better at utilizing the different substrates. So we have some ideas that a higher diversity in general terms is a good sign. It's not a universal truth, because we also know that in adults, you can have high diversity due to being constipated. So we know that the transit time, the time it takes food to travel through the system, is strongly related to diversity as well. So it's not a universal truth that high bacterial diversity is associated with health, but generally speaking, that it is a good proxy of a diverse diet and a healthy lifestyle.
And when does the gut microbiome of a child stabilizes? Because it goes from, being sterile at birth to this colonization process. So what happens then?
I think most studies would suggest that the microbiome of a child starts to establish around 3-4 years of age, maybe a little later. I don't think we have really good understanding of what is ultimately determining stabilization. My own ideas is that it really depends on to what extent the child has fully transitioned to a family diet, which is complex and diverse. So some children will still, at 1 year of age, eat a very simplistic, simple diet with maybe not a lot of vegetables, whereas others, they would more rapidly adhere to a diet that is more similar to an adult microbiome. So ultimately, I think the complex diet when they have solid foods would bring more stability to the microbiome faster than a more simple diet. So I think that's probably a part of their equation here.
And does the gut microbiome become more established at some point?
Yeah, so I would say that when the child is 3, 4, 5 years age, then it's more established and less variable. And actually, our microbiome in adults is incredibly stable. So it's actually really hard to change your microbiome. It requires drastic dietary changes, and it requires sustained drastic dietary changes. So if you change your diet for a week or two and you stop, you'll go back to your original composition very fast within a few days. So that also suggests that actually the microbiome we're born with and that we develop through the first year of lives could be important for setting your microbiome also in adulthood.
Yeah, it could mean potentially that what happens in those years sets you up for a good microbiome and potentially health as well, but also preferences perhaps for different types of foods.
Potentially, yes. I don't think that's fully understood at this point in time.
Yeah. And what are the other factors that affect the gut microbiome? For example, a lot of people ask us about antibiotics.
Yeah, so we know antibiotics, their purpose is to kill bacteria. So for sure, if you consume antibiotics, that would have an effect. I don't think it's a problem with a few antibiotic treatments here and there. The problem is really if you have multiple long-term antibiotics treatment, then you put additional stress on the microbiome, and that could have detrimental effects that the microbiome would not reestablish itself after treatments. So it should be used with care and attention to only using it when it's really needed.
Of course, antibiotics can be life-saving and can be
for sure.
needed in many situations. Are they all the same, or do different antibiotics have different effects?
Yeah, so some antibiotics are very narrow-spectred. So that means that they are targeted certain types of bacteria, where others are more broad-spectred, targeting a range of bacteria. And of course, we want to avoid using the broad spectrum antibiotics as much as possible to basically don't harm more than necessary.
And antibiotics have also been associated to some health conditions later in life, especially if taken early on.
Yeah, so I think there are evidence that multiple antibiotics treatment early in life can have or is associated with later health implications like allergy or eczema. And there is also suggestions that antibiotics treatment early life maybe also affect propensity to obesity later in life. But again, it's associations studies, and it's hard to know whether there could be other lifestyle-related factors that could explain part of the effects that are seen.
What do we know about diet and how it affects the gut microbiome?
So I think, as we discussed before, I mean, diet is ultimately dictating what kind of substrates you feed your bacteria with. So if you eat a diet absent of any dietary fiber, you're basically starving your microbes because all the nutrients will be absorbed in the small intestine and very little will end up in the large intestine and being available for the microbes, which are mainly populating the large intestine. On the contrary, if you eat a diet that's diverse, full of dietary fiber that our own digestive enzymes cannot degrade, then you are feeding your microbes to a much larger extent. So there is a direct link between what we eat, which microbes are there, and also in particular the activity of the microbes, because production of molecules that microbes produce are ultimately dependent on the substrates that are available for their growth and their metabolism.
Are there some foods that could be bad for the gut microbiome?
So I think that's a very simplistic view to say whether if any food is bad, but everything is about balance, right? So too much of one food could be bad. So if you only eat ultra processed foods totally absent of any fiber, full of sugars, then too much of that would harm your microbes. But I would not say one single food is bad. It's all about balance. You can also say meat is not bad per se, but if you only eat meat, then you're also starving your microbes because you're basically not feeding any fibers to the microbes. If you keep your fiber intake high, then meat is perfectly fine, and it provides a lot of different amino acids that are useful for your body as well. So it's always a matter of balance.
Is there something in ultra-processed foods that affects the microbiome?
I think the problem with ultra-processed food is probably the lack of certain nutrients from a microbiome perspective. So it's the lack of dietary fiber, the lack of whole grains, the high availability of sugar, which would never reach the large intestine. And that's the main problem from a microbiome point of view. Many ultra-processed foods are very energy-rich, full of sugar and salts, and too much of it would not be good. The problem, however, with the term ultra-processed foods is that it does not necessarily say anything about the health or nutritional content of the food itself. It only refers to how it was processed. So, for example, in Denmark, where we like to eat rye bread, so rye bread would be considered a ultra-processed food, although that we know that there's for sure a lot of health benefits of the food. So we have to be careful when we talk about ultra-processed foods as being the bad thing. It really depends on what we talk about.
Maybe generally ultra-processed food, or it's easier to preserve and store food when you remove the fiber. So white bread, white flour, whenever you remove the fiber, it's cheaper, easier, and has a longer shelf life, or at least that's been kind of the practice in the past. But as you say, there's different types, and you always need to look at
what are we talking about?
What we're talking about. And it seems like fiber is really here, the key word.
And diversity of fibers as well.
Yeah. What about sugar? Does it have an impact on the gut microbiome?
So if you eat sugar, most of the sugars will be absorbed in the small intestine. So will never reach the large intestine. So in that sense, you're starving your microbes again. So I think it's more the lack of fiber per se.
Okay, so fiber is really the key word here, and the diversity.
Yeah, a varied diet basically is the key here.
Exactly.
Yeah.
And maybe the way to visualize, I really liked your metaphor. In a way, we're talking about substrate, so like food for microbes.
Yeah, exactly.
So the more different ones you have, the more microbes you're feeding, and the more diverse your gut microbiome.
And then I think it's also important to realize that it's not only about the microbes when you eat a diverse diet full of fiber, it's also about stimulating gut motility. So contractions in the intestine needs to be stimulated. And part of that is a physical stimulation by the food. So if you have a very liquid diet, it would not stimulate contractions. If you eat more vegetables, which are less easily digested, they would, to a larger extent, stimulate contractions in the intestine that will help the food to pass through the intestine. And that's important for regular bowel movements and for avoiding constipation, basically. And I think regular bowel movements are super important for well-being, and I don't think that should be neglected as well. So it's not only about the microbes, it's also about stimulating the gut physiology and the functionality of the intestine.
And we said that no bacteria are bad per se, but there are some bacteria that in high amounts could be potentially unfriendly.
Yeah.
What do we know about that? And are there any bacteria that we could consider bad or unfriendly, we could say?
So I think in a healthy individual, you can easily have unfriendly pathogenic bacteria being there without harming you. But the problem is that if your barrier function in the intestine is suddenly compromised, then these bacteria might have certain components in them that can sort of stimulate the immune system and give rise to an inflammatory response. So certain bacteria have sort of features that the immune system recognize and would react to. So that's why that certain bacteria could more easily stimulate the immune system than others.
Yeah. What are those bacteria? There are also some that are associated to some infections or conditions. What do we know there?
Well, for example, we have certain E. coli that could be problematic, but again, we also have a lot of E. coli that are non-problematic. So it very much depends on the strains of the E. coli, whether they are potentially pathogenic or not. So this could be one of the, or Klebsiella or certain bacteria that sometimes cause troubles. And what's characteristic about these microbes is also that they typically don't tolerate low pH. So if you don't have a lot of Bifidobacteria to keep pH low, then maybe you have higher pH in the intestine, and suddenly these bacteria can start dominating, and some of them grow really fast, and then they can take over a lot of their community in the gut, and that can also be a problem potentially for having a stable ecosystem.
And still talking about association, in our study, the PREVENT study, we found an association between the gut microbiome in children and constipation. Is that something you have also observed?
So we have not in our infant cohort studied this yet. We will study it soon, but we know from our studies in adults that there is definitely a link between constipation and composition of microbes and also activity of microbes. So we recently did a study in adults, which were a little more invasive than what you will do in infants, where volunteers swallowed a smart pill, which is a pill with electrodes that can measure pH, temperature, and pressure as it travels through the GI tract. And what we learned from that study is basically that by measuring pH in the different segments of the intestine, we could see that people are very different when we compare the pH from individual to individual, but also that the time it takes the smart pill to travel through the small intestine and large intestine is also highly variable. So that's basically what we call transit time. And we could see in that study and also in previous studies that transit time, so the time it takes food to travel through the system, is strongly correlated with the microbiome composition and activity. And if you have constipation, you basically have a very long transit time.
Yeah.
So you're finding that transit time correlates with bacteria, or the constipation correlates with bacteria. Makes perfect sense from what we know from the adults.
What are the other characteristics or conditions you will be looking into in the motility cohort? You talked a bit about it before.
Yeah, so defecation patterns, early life nutrition, so transition from breastfeeding to solid foods is something we're very curious about. We would also like to understand how the children's growth, whether it's in any way related to the nutrition and to the microbes. And then immune development is something down the line that we also hope to understand better. Yeah, for sure.
Exactly. You also have blood samples and other types of samples from the cohort. So you'll be able to put together a lot of different pieces of the puzzle, it seems.
Yeah. Yeah, we both were very fortunate that parents volunteered to have a bit of blood taken from their child. And that will enable us to study the immune cell population at 3, 6, 9, and 12 months and see how it changes with the development of the microbiome and with the transition and diet. And then we also collected urine samples using the diapers. And there we have monthly urine samples throughout the first year of life. And when we study the molecules in the urine, we can sort of both track changes in microbial activity, because certain molecules from the gut are being absorbed into the blood and eventually excreted into the urine. And we can actually also, the ambition is also to track dietary patterns in the urine, so we can get more objective data on the children's diet by studying the molecules originating from different foods in the urine. So then we can hopefully also see their trajectories in dietary patterns during the first year of life being reflected in the urine. And we hope that can help us also understand better the development of the microbiome.
So this is not just looking at the microbes, but also what they're doing, what they're producing.
Exactly.
And how the rest is also affected.
Yeah, so in our group, we have a very strong interest in understanding the microbial metabolites, the small molecules the microbes produce, to figure out which of these are there in abundance, which microbes produce these molecules, and which molecules are able to affect gut motility or the immune cells. Because ultimately, I think that's where we need to go. We need to not only understand which microbes are there, but more understanding what do they do and how can they affect our metabolism, basically. Because only once we know that, then we can better sort of target the right pathways. We can better understand how can we shape the children's health in early life. And that's ultimately where we need to go.
Yeah, exactly. And it seems like it's so complex. There are so many things acting together and so many things to be studied, but there's also a lot of variability from one person to another.
Yeah.
So one of the challenges we also encountered in our study is that you need a lot of data and you need a lot of people participating in the study. You need to follow them over time. You need a lot of samples and you need to ask also a lot of things.
Yeah.
And it's also very difficult to study diet because, as you said, it's very difficult to ask if a child introduced solids, because what do we mean there?
Yeah.
Do we mean they just tasted something, or do we mean it's their main source of nutrition?
Yeah.
So there's a lot of data we need to advance this field. What do you think? How do you think we can collect more and more data, or let's say put these data sets together? I think that's the main bottleneck at the moment.
I think one thing is that we work with the longitudinal aspect. So that's why we, instead of looking across individual, we would rather look for changes within an individual over time, because that's one way to get a better understanding how things are related, because we study within children over time. So that's one aspect. But I think it's very important to, once we see associations in the children, that we actually work backwards to understand the mechanisms. So that's why we collaborate with outstanding scientists to figure out, once we see associations between a given microbe and a molecule, then we test, okay, can this microbe actually produce that molecule? Then we show that, yes, it do in fact produce it. Then we try to understand, why does it produce a molecule? We identify the genes and the enzymes responsible. And then we go further and say, okay, can this molecule actually affect the immune system? And then we work with immunologists that can test in controlled experiments what happens if you expose immune cells to this molecule. Does it have an effect? And if it does, how? And I think that's really where we need to go. Not only finding patterns in the infants, but really use their patterns in infants to work backwards to understand, why is it that we see these links, and are they meaningful? Does it mean anything? And then, of course, we need to see whether we can replicate our findings across cultures, across different geographical locations, etc.
And the longitudinal aspect is extremely important, because especially in trying to understand if someone is healthy is very tricky.
Yeah.
Whenever you collect data from any subjects and you try to understand which ones are healthy or not, it's very difficult, because you don't know what will happen to those people in a year time. But when you follow children over time, you have more and more data on what will happen afterwards. So that's a big advance, in my opinion, also, to not just compare or look at standard population, but looking at people over time.
Yeah.
And I think in the Nordics, there's so many interesting cohort studies. I would say Denmark, Sweden, Finland have a lot of big cohort studies that follow people, especially children, over time. So it's fantastic to have this.
Yeah, and that's very important, because if we really think that what goes on in the intestine early in life sets us up for life later, or for risk of diseases later in life, then we ultimately need to, first of all, see the links between early life and later disease, and then work backwards and try to explain, if we see these links, how can we understand this? And if we are then to actually prove that we can do something about the risk in early life, we actually also need to do an interventions early in life, where we try to affect the risk for later disease. And that takes time, money,
Yeah, but would be incredibly valuable for society.
Definitely. Yeah,
once
it would be really valuable
to know more about this, because this could quickly affect many generations ahead.
Yeah.
Awesome. I think we've covered a lot of topics. Thank you so much, Henrik, for explaining the gut microbiome in early life, in adulthood. This was very, very interesting. Thanks a lot.
Thank you very much.